Hello, my name is Julia Harencar, and I'm really excited to be sharing my work on closely related ginger species remaining distinct despite recent and ancient gene flow. So introgression is just realized gene flow between two species, and it's actually really common across the tree of life. We've seen it in our own human lineage with Neanderthals and Denisovans. We've seen it in certainly many different plant systems, in other vertebrate systems like salamanders, and invertebrate systems like butterflies. It's pretty ubiquitous across the tree of life. And many of these cases, those species are maintained as distinct despite all of that genetic sharing. And I'm really intrigued by this question of how is that possible? And what are the mechanisms, both genetic and ecological and evolutionary, that maintain that distinction? And so to answer this question, one of the first things we do is look at the nature of the hybridization or the introgression that we see between our species of interest. So how far does introgression extend into the species, both geographically and through time? So is there both ancient and recent introgression, or is there only recent introgression? And then how does adaptation and selection inform patterns of introgression, both across the landscape and across the genome? So today I'm going to answer these first two questions for my system and talk a little bit about my ongoing research on this third question. So I study these questions in the Neotropics, which are an excellent place to do so. They are the most diverse place on Earth, and the understory plants in particular are full of recent and very, very rapid species radiations that we still don't really understand super well. Particularly, we're not totally clear on how so many close relatives are able to coexist, especially because with plants in particular and recent radiations, there is the ability for all of these species to interbreed. They're interfertile across the whole genus. So I look at this question specifically in villosissimus and allenii, these two species here that are not each other's sister species, but are very closely related. They've only diverged in the last roughly 1 million years. Costus allenii's range is entirely held within Costus villosissimus's range in Central America. And yet locally, they partition habitat differently. So allenii grows in perennially wet forest understory, whereas villosissimus grows specifically in much more open and often seasonally dry forest edges. This differential habitat adaptation is very much a part of their reproductive isolation. So it was previously identified as one of the key reproductive isolating barriers between the two species. And you can see from this reciprocal transplant figure with fitness on the y-axis and habitat on the x-axis that allenii basically can't survive in villosissimus's habitat and vice versa. So really strong habitat adaptation, differential habitat adaptation. And yet they're able to readily hybridize. So these are hybrid individuals from the greenhouse, but they also share pollinators, these really cool orchid bees in the wild. And so it's likely that there's wild hybridization occurring, especially in places like Panama, where they occur in really close proximity. And this is where I study these two species. The precipitation gradient across the Isthmus of Panama is really steep, such that the northern part of the isthmus receives perennial rain, whereas to the south we have a 3 to 4 month dry season. And so we get primarily villosissimus to the south, and we find a lot more of the allenii to the north. But right in the center of that precipitation gradient, right along the canal, we can easily find hybrids, or what appear to be hybrids. And so I wanted to know, first of all, are these actual hybrids? And then what is the extent of that introgression, both across the landscape and a little bit through time? Are they only recent hybrids, or are they advanced generation hybrids as well? Without getting into the nitty-gritty of my methods, because we only have 8 minutes, I'll just say I did low coverage whole genome sequencing and then analyze that data in a probabilistic framework using angst. Happy to talk more about that. But for now, let's jump into the PCA here, the first result. On the PC axis 1, we have the separation of allenii and villosissimus ancestry with really clear hybrids right there in the middle. And what we found as the genetic evidence for what hybrids were corresponded fairly well with what we call this hybrids in the field. Though it turns out allenii and the hybrids look a lot more similar than villosissimus and the hybrids. And that slightly extended the area that we thought was the hybrid zone because we found some hybrids where we thought there was only allenii. And again, I wanted to ask next, are these only recent generation hybrids? Are they F1s, or have they been experiencing backcrossing and more advanced generation hybridization? So if we look at one individual from this PCA, one hybrid, I then did a hidden Markov model ancestry analysis. So in this figure here, the chromosome position is along the x-axis, and the y-axis represents ancestry, with 1 being individuals homozygous for allenii ancestry, 0 homozygous for villosissimus ancestry, and 0.5 heterozygous between the two. So really clearly, this whole chromosome 1 for this individual is heterozygous, indicating that this is an F1. So mom slash dad was allenii, and the other parent was villosissimus. I also found some individuals that were clear back crosses. So F1 parent, we see chunks of F1 ancestry, and then through recombination, chunks of villosissimus ancestry. And therefore, right, mostly we see recent hybridization in these individuals. And I wanted to know whether it's possible that the anthropogenic history of this land is the reason for this hybridization. So Pipeline Road was constructed in World War II to bring oil across the isthmus. And because of that, it creates a intermediate habitat where we have high light and a high light environment running through this precipitation gradient. And so I wanted to know, is there also ancient introgression between these species that would precede the construction of this road and this hybrid habitat? So to do this, I did ABA-BABA analysis. I won't get into the nitty-gritty of how this works, other than to say that there are 2 different gene genealogies that can arise from incomplete lineage sorting. And we basically expect those to occur in equal frequency when there's no introgression. So by neutral processes, we should see these patterns roughly equally. To make sure that I didn't deal with reference bias, I also sequenced a new genome, the Costus genome, a little COVID project. So that is a big help for these analyses, as we don't have to worry about reference bias. And then if there is introgression, we really expect that the ABA genealogy is going to be more common because of sharing of genetic information between allenii and villosissimus through that hybridization. This is exactly what we see, far more ABA genealogies than BABA genealogies. And it, in reality, makes a lot of sense because of how much the area over which these species co-occur, much of which is not heavily influenced by anthropogenic disturbance. And so in summary, we see both recent and ancient introgression, and it's likely that this really strong habitat adaptation is what maintains distinction despite this ongoing gene flow. So my current work is looking at patterns of introgression across the genome and trying to align that to a quantitative trait locus analysis that I'm conducting to map the regions responsible for key differential adaptation, with the expectation that regions with less introgression are going to correspond to those regions critical for maintaining reproductive isolation and differential habitat adaptation between these species. With that, I want to thank all the people that made this work possible, especially my 2 Indigenous field assistants. They've taught me so much about the neotropics, where to put plants, where you can't put plants, and taken excellent care of my plants when I was unable to go down. And of course, to the many undergrads, my committee, et cetera, funding sources, for everything that they have done to make this possible. And thank you for listening.